we have the highest percentage of Black and brown populations, and also the greatest incidence of poverty, which coincides with poor nutrition and increased vulnerability to contaminant exposure. Next slide, please. In 2017, I met County Commissioner Alan Booker with Rebuilding Together and Doris Dollar Carter. They're both involved with EJAB. And in speaking with a lot of residents, we learned that there is a lot of fear and misinformation in the community about seafood consumption advisories. And a lot of the information that is accurate is very difficult to access. And so we wanted to identify an awareness vehicle that brought a more respectful and celebratory light to our traditions as coastal consumers of seafood. And so we decided to use a cookbook as the food tie that binds, where residents contribute recipes that can be consumed in line with the consumption advisories. Next slide, please. So we currently are on our third version. This is going to be a dedication to local seafood houses and restaurants and Black-owned small businesses. Version 2 is available in hard copy through EJAB. In 2019, the first version is available online for a free download. Next slide, please. So to give you some quick visuals, we have in structure of the cookbook, we have background on marsh ecology, so our home and our habitats. And with each recipe, we also have a biofact included. And we also invited our contributors to submit a small testimonial about what the recipe or seafood or Brunswick means to them, so that we could also make this be a tribute to our local residents. Next slide, please. Secondly, we have our fisheries and the seafood species that are included in the advisories. Next slide, please. And then last, we have the advisories themselves that are coupled with consumption and preparation guidelines. And then we have the full tables. Next slide, please. And as you can see, the tables are also color coded along with the sampling zones on the map that are associated with the advisories. And we do have all of the advisories for Glenn County. So it is a comprehensive county resource. So thank you for attending my cookbook drive-through. And I'd love to hear questions or suggestions. This is an ongoing project, and we're really, now we're in the stages of getting feedback from the community about how this is working. So we'd love to hear your thoughts as well. Thanks so much. Next up, we have Kristen Archer, who is presenting on Grace Reef. Kristen.
Good morning, everybody. Can you guys hear me okay?
Kristen, yes. It looks like you're unmuted, but we cannot hear you.
You guys cannot hear me at all?
No, we can hear you.
I can hear you just fine.
Okay.
I can hear you.
All right. Thanks, guys. So good morning, everybody. My name is Kristen Alexi Archer, and I am the Georgia State Sea Grant Fellow for Grays Reef National Marine Sanctuary. Next slide, please. So here at Grays Reef, we do stationary mobile acoustic telemetry using acoustic receivers and hydrophones to hear sound and general communication in Grays Reef. So we have 16 receivers spread across the sanctuary, and so we'll tag different species of fish, and then when those tagged fish get near the acoustic receivers, it gives off a signal to those receivers that collects that tagging information so we understand species movement. We also have 3 hydrophones that are coupled to 3 of those acoustic receivers throughout the sanctuary, and that helps us hear general sound. So we'll hear fish communication, other organisms making sound, as well as boat noises, thunderstorms, and wind. So it helps us get a general idea of the sanctuary soundscape. Next slide, please. So why do we monitor sound in the sanctuary? It's essential to understand changes in the sanctuary overall, so it'll help us understand what communications and sounds are these animals making during day and night versus different seasons versus different years, and then general movement for these animals and where they're going, how much time they're spending in the sanctuary, where they like to be. It also helps us monitor important species movement, so black sea bass, red drum, scamp grouper. And then we also monitor sanctuary use, so understanding how many small boats are out there, how many divers are out there, and understanding what type of anthropogenic noise is impacting these organisms that are within the sanctuary. And then we also use that to understand what type of environmental noise there is, so thunderstorms, wind, and just overall understanding what the soundscape at Gray's Reef sounds like and how that impacts the organisms and what sounds they're making as a whole. Next slide, please. So why is it important to the broader coastal environment? So overall, it helps us develop a telemetry network across the sanctuary, but more importantly, across the southeastern United States. So there's a centralized database of information for these acoustic receivers called the FACT network, and it allows us to share information from our acoustic receivers. So if the species we tag at Gray's Reef go to other places, we can get the tagging information from those acoustic receivers to understand how much they use the sanctuary versus the other places they go. Likewise, other projects working on highly migratory species, when they go through Gray's Reef, they can also collect that information, allowing us to have an overall telemetry network across the southeastern United States. Similar to this, the Navy has a sound project with the National Marine Sanctuaries, 3 specifically on the eastern United States. So Gray's Reef, Florida Keys, and Stellwagen Banks National Marine Sanctuary all have hydrophones from this Navy project. And it allows us to understand environmental sound and then how there are different soundscapes depending on the different noises impacting those sanctuaries. So for instance, Gray's Reef and Florida Keys have a lot of small boat traffic, whereas Stellwagen Banks has to deal with bigger boats because of the shipping lanes. Overall, it also helps us to monitor climate change, so we can understand the movement of these species and sounds they make during the current environmental conditions, and then as those conditions change over years, as the climate changes, we can understand how that might impact their home ranges, how it might shift, how it might increase or decrease, and understand it as a whole over time. Thank you guys for your time.
Kristen, do you have any bonnethead sharks that are tagged?
We don't tag any, but we have picked up different sharks on our acoustic receivers, but I can't tell you the species off the top of my head.
Okay, thanks.
Thank you so much, Kristen. Next up, we have Mark Hough. And I do apologize. We had an issue with the slides, but I think they'll work out. So, Mark.
Hello, I'm Mark Hough. I am a research technician for Marine Extension Georgia Sea Grant in the Coastal Ecology Lab under Kimberly. And I'm going to be talking to you about genetic relatedness as a driving factor of social interactions in American alligators. Next slide, please. So what we're doing is we're basically comparing genetics in relation to movement, territory, behavior. So how the genetics affects those 3. So for an example here, you can see on this map, we have the ranges of female alligators. And for the most part, they're separate and they keep to themselves. But if you notice, the white and purple circles are inside the blue and orange circles there on the right side. So what we're hoping is that when we can test for genetics, we can determine if that is playing a factor. Are they more related? Is that why they're allowing them into their territory? Next slide, please. So how we're doing this, we are collecting tail tissue samples from as many alligators as we can so we can put together a DNA base essentially, and we can compare and see what alligators are related to each other. And then we're going to relate that back to the satellite tags we have on adult animals, as well as the game cameras we have deployed in the swamp. We're going to look for patterns to see if they're the more related alligators or the less related alligators are doing similar things, and if that's playing a factor in their behavior and movement and all that. Next slide, please. So why are we doing this? The health of the alligators and their populations are tied directly to the health of the ecosystems. Alligators are a keystone species, so they play a major part in maintaining the ecosystem. They're ecosystem engineers, so they modify the ecosystem to help other animals. So on the top picture, you have a gator hole, which during drought conditions, that's one of the last sources of water remaining that fish and other animals will use. And then on the bottom is a gator den, which abandoned gator dens are used by other animals for habitat. And then on top of this, they are apex predators, as most of you know. So they help maintain all the mesopredators that would otherwise run wild and could cause other problems. Thank you, and my email is at the bottom there if you guys have further questions.
Musical computers. Thank you very much, Mark. Next up, we have Heather Jousting. My apologies if I mispronounced your last name, Heather.
Oh, no problem. So hi, everyone. I am Dr. Heather Jousting. I'm an associate professor at Georgia Southern University on the Armstrong or Savannah campus. And my overall work is focused on coastal plant ecology. And my overall interest has been in using native plant species in restoration projects and increasing diversity of things like the plants we use in sand dune systems. I've done extensive work in the past in sand dunes, but recently I have moved my interest into the salt marshes. And as you guys know, Georgia salt marshes are one of our really key resources here in Georgia, and it covers near 360,000 of coastline. And we all know the critical ecosystem functions that it provides, including the basic nurseries for fish and shellfish. And so I've been interested in using our native plant species, Spartina alterniflora, in living shorelines. This is commonly planted as part of the restoration projects. And it's due to the important role that Spartina plays in the marsh. It functions to not only provide the base of the net primary production that supports and makes these systems so productive, but it also plays a critical role in marsh growth and maintenance. The upright growth of the plant species attenuates the wave energy and allows for sedimentation to occur. And the roots, of course, provide stabilization that helps the marsh grow both vertically and horizontally. Next slide, please. So what we've been interested in is that locally there is no Georgia source for native Spartina for restoration projects. When these restoration projects occur, most of this material is purchased from out of state. And studies have shown that there is, because due to the wide range of Spartina and occurring from the Northeast down through South America, there's a lot of genetic variation within Spartina where you have local adaptation to native habitats. And so Georgia has a very unique coastline. We have a very unique tidal range. And so I have been interested in trying to create a source for Georgia native plants that can be used in restoration projects in Georgia. So the overall theme here is to determine the best practices for producing native Spartina. And currently, my lab has been looking at both characterizing the phenology and the fecundity of Georgia marsh populations, and also different novel ways to grow and nursery produce these plants. So we've done some field work in collecting data on flowering status and reproductive status, as well as the collection of Spartina seeds from various marsh habitats and looking at things like seed count, seed viability. We also have been looking at germination success and survivability. And we're also interested in trying to look at producing Spartina in an above ground wetland that has been designed to remediate aquaculture effluent. And so I don't have a picture of the marsh or the above ground wetland because we're currently completing building. It's pretty unimpressive in its pictures right now, but the overall design of the schematic is shown below. And so we're wanting to use basic aquaculture to connect this remediation wetland and look at both the ability of the wetland to produce Spartina. We're going to compare that to traditional methods, and also the ability of the Spartina to remediate the aquaculture effluent. And so we're hoping to eventually, right now we're using a freshwater species, but we're hoping to, in the future, use a marine aquaculture produced species so that we can alleviate some of the pressures on remediation of water for aquaculture. Next slide, please. We also, I just have some preliminary data below on some of the trends that we've seen in Spartina, phenology, and fecundity. We looked at kind of the dates, best time period to collect seeds, which we've found kind of in October-ish. And we've looked at seed viability with different marine influences. And so the sites below just have I know there's no descriptor there, but the sites below are varying distance from the ocean and have varying salinity. So we've been kind of looking at where's the best place to collect viable seeds and when's the best time. I also just received a coastal incentive grant for this cycle with my collaborator, Joel Koska from Georgia Tech, to continue this work and trying to determine some of the best practices for nursery production. So we're gonna kind of look at some variation in genetic variation within nursery produced Spartina, as well as wild populations. And we're gonna look at kind of coming up with the best way to germinate and store in cold stratification like time periods to have best seedling survival. And we're also gonna manipulate that wetland a little bit to more represent marsh conditions with tidal flow to kind of look at the optimal source material for restoration success, transplants versus seedlings. And so the overall goal here is to come up with some protocols for cultivation and kind of jumpstart this nursery production of Georgia Spartina. And that's kind of it.
Thank you very much, Heather. We're going to need to wrap up.
Okay.
Move on to our next speaker. Thank you so much for your presentation and your amazing work.
Thanks.
We're going to finish this first session of the lightning round with Kumar.
I don't think Kun is with us. Yeah, she doesn't seem to be with us. Kimberly, so is it okay if we move on to some introductions?
Absolutely.
Great.
Thank you all for the presentations.
Thank you so much, Kimberly. Good morning, everybody. We'll move on to some introductions. Want to make sure that everybody gets a chance to introduce themselves. So let's see. Yesterday, we went from A through E. We do have some people who didn't get a chance to introduce themselves. So Chandler, would you unmute yourself, tell us your name, title, affiliation, and your research interests, please. Chandler, are you with us?
Yeah, sorry. My name is Chandler Calmer. I'm a student at the College of Coastal Georgia. I'm also interning for the Georgia DNR Coastal Headquarters, and I'm also interning for the UGA Merrick's. Right now, I'm trying to learn, different projects that other departments are doing. So I think this symposium will, I get to see a few of the projects that other departments are doing.
Good to have you, Chandler. Thank you for being here. Isaac Murphree. Okay, moving on. Jackie Kelly.
Hey everybody, I'm Jackie Kelly, Associate Professor of Geology at Georgia Southern University. I apologize for the background noise. I'm a geochemist by nature, so I study groundwater discharge along the coast. It's nice to see all of you this morning.
Good to see you, Jackie. Sorry, I mispronounced your name there. Moving on, Jan McKinnon. Okay, Jeb Byers. Moving on, we'll go to Jennifer Dorick.
Hi, I'm Jennifer Dorick. I am a first-year PhD student in the food science department. You heard my presentation yesterday. I am on the 2020-2021 Georgia Sea Grant traineeship, and I'll be continuing my traineeship the following year to continue my research.
Fabulous. Thank you for being here. Isaac?
Hi, everyone. My name is Isaac Murphree. I'm a Marine Education Fellow with UJ Marine Extension Georgia Sea Grant out of the Marine Education Center on Skidway Island. I'm really interested in all the work everyone's doing, but especially anything to do with reptiles. I'm a big herpetology person, so it's really cool to see some of the work that's being done specifically out of Dr. Andrews' lab and some other places as well. So happy to be here.
Great, good to have you. Jeb, are you with us?
I'm here.
Ah,
I'm here. Hi, I'm Jeb Byers. I'm a professor in ecology at UGA. Broadly, I work on marine population and community ecology. Some of the specific areas I work in are number 1, invasive species, number 2, disease ecology, 3 is ecosystem engineers and the foundational species, and 4 is looking at species range boundaries, particularly how they're changing with climate. Some of the specific projects we're working on have actually been touched on by some of the other speakers who I'm collaborating with, like Mark Fisher. We're working on the black gill disease in shrimp, and John Carroll mentioned some of the oyster disease work, which we're getting up and running. We've also been working extensively on the invasive red seaweed Gracilaria vermiculophylla, which you may have seen all over the lower intertidal reaches of the mudflats in our area. So those are just a few snippets of some projects that we're actively involved on. Thank you. I've enjoyed all the sessions this week.
thank you so much for being here, Jeb. Moving on, Dr. Joel Koska.
Hi, everybody. I'm Joel Koska. I'm a professor and associate chair for research in biological sciences, and I'll be talking next hour, so I won't take up anybody's time.
Thank you so much. Jose Rolando.
Hi, I am Jose Rolando. I'm a PhD student in Joel Koska's lab. I'm basically been working in microbial plant interactions, especially in Spartanatony flora and in the root zone. And I think more lately also shifting into doing more broad scale ecology in salt marshes in general, moving more into the ecosystem scale.
Lovely, good to see you, Jose. Jose was also one of our Sea Grant trainees. It's always nice to see our students. Moving on to Katie Smith. Katie?
Hey there, good morning, everybody. Really enjoyed these last few days with you all. I'm Katie Smith. I'm the Water Quality Program Coordinator for Marine Extension and Georgia Sea Grant in our Brunswick office. Started my career with Marine Extension, doing a lot of nutrient and bacterial work in our estuary. In recent years, I've been really involved in a lot of marine debris education. I serve on the Consumer Debris Reduction Work Group with NOAA Marine Debris Program. Got a freshwater wetlands project that I've been working on for the last few years. It involves educators, students, college students, as well as K-12 students. So kind of just dabbling in the water resources realm. I serve on the water resources visioning team for the Sea Grant. So just enjoy connecting with researchers, hearing about all of the amazing work. I'm always so just mind blown by just the wonderful researchers that dedicate their career to studying our wonderful Georgia coast. So just excited to see where, how far everybody's come, and just I really enjoy, trying to pick out pieces that we can, help just enhance our education and outreach efforts, and look forward to hearing the rest of the presentations today. Thanks.
Thank you, Katie. Karen Giovango.
Hello, everyone. Good morning. I'm Karen Giovango. I'm Program Manager of UGA Marine Extension and Georgia Sea Grant's Ecoscape Sustainable Land Use Program, which entails encouraging responsible stewardship of our natural resources. I emphasize native plants and provide research support, as well as technical assistance, education outreach, and development of different resources to assist both professionals and non-professionals. Examples include drafts of professional native plant guidance regarding bioretention systems for coastal Georgia, applications, resilient salt tolerant native plant applications as well. And I'm very interested in several of you researchers regarding interest to work with you all a broad strategy for maximizing native plant material diversity for both ecological restoration, germplasm, conservation, et cetera. So I'm really looking forward to the discussions today. And if you're interested in any way discussing further with me, I look forward to it. Thank you.
Thank you so much. We never got a chance to meet with Courtney Mcgill. Courtney, would you unmute yourself and introduce yourself, please?
Hi, yes, my name is Courtney Mcgill. I'm the program specialist here at University of Georgia Marine Extension in Brunswick. My research interests vary. I'm just interested in all the multiple topics that have been discussed, and I'm interested to see what all of our researchers have been working on.
Lovely. Good to see you, Courtney. Thank you. Moving on to Kevin Haas. Kevin?
Hi, I'm Kevin Haas. I'm on the faculty with Civil Environment Engineering at Georgia Tech, as well as the co-director of our joint PhD program in Ocean Science and Engineering. I do a lot of research in coastal engineering, a lot of numerical modeling and field measurement of coastal circulation, tides, ship wake, various different coastal engineering impacts. I also spend a lot of time working on ocean energy. So that is using tides, waves, and currents for generating electricity. So thank you.
Good to see you this morning, Kevin. Moving on to Kimberly Robertson. Kimberly?
Yes, good morning, everyone. Thank you, Mona. I am Kimberly Robertson. I work at Grays Reef National Marine Sanctuary as the research coordinator. I'm very happy to be here learning about the research in the area along coastal Georgia. Our site, Grays Reef National Marine Sanctuary, lies 20 miles offshore. You just heard Kristen Archer talk about some of the research that we have ongoing there. We also partner with a lot of folks in the region that you have heard from today, including Danny Gleason, Mark Fisher, others, and looking at the invertebrates, looking at ocean acidification, climate change, how it affects, potentially affects the site, studying live bottom and the connectivity of Grays Reef to the South Atlantic Bight and beyond. As the research coordinator, I'm also looking for opportunities for those interested in those topics and others to work at Grays Reef. So if you are interested, please do feel free to contact Grays Reef National Marine Sanctuary and me. Thank you.
Always great to see you, Kimberly. Thank you. Lisa Leach. I hope I said your name right.
Good morning. I'm Lisa Leaguey. I'm a professor of biology at Georgia Southern University on the Statesboro campus. I am a plant ecologist. I work with rare and endangered plants. And more recently, I've gotten back into coastal work. I've done some work on coastal sand dune ecology and restoration. And most recently, I've gotten a coastal incentive grant with the city of Tybee to work on coastal dune restoration there with the reconstructed sand dune. And my graduate student, Shannon, who I'm sure you'll be hearing from soon, she's a Sea Grant research trainee this year. So it's been an exciting year to get back involved with coastal work. Enjoying hearing about what everybody's doing out there. I also want to mention I'm also an environmental science textbook author, and I'm always looking for great examples to put into our updates in the textbook. So thank you.
Lisa, thank you so much for being here. I miss seeing everybody in person. Moving on to Lizzie King.
Hi, good morning, everyone. I'll be talking in the next section, but I'm at the University of Georgia, interested in restoration ecology and social ecological systems. So we'll talk to you in the next section. Thanks.
Look forward to learning more from you, Lizzie. Thank you. Moving to Luke Robertson. Luke.
Hello, my name is Luke Robertson. I'm the Adopted Wetland Coordinator here at UGA Marine Extension, Georgia Sea Grant here in Savannah. And I often pair with researchers, teachers, and volunteers, students, and the like to learn more about water quality on the coast and then become coastal stewards by getting them out and sampling the waters near their neck of the woods. I'm really enjoying the talks, and I'm looking forward to being a moderator in the next session later today. So I'll see y'all soon.
Thanks for all your hard work, Luke. Thanks for being here. Moving on to Marshall Talford. I hope I said your name right.
Yeah, it's fine. Good morning, everybody. I'm Marcie Fair. I'm a geochemist and a Georgia Tech professor in Earth and Atmospheric Sciences. And in my group, we work mostly on sediment biogeochemistry. interested in the carbon cycle. And today you'll hear more about our project with Sea Grant, which is the first time we're involved with Sea Grant actually. So it's pretty exciting to be here and listen to all these thoughts. Our project is on ocean acidification.
Very much look forward to learning more from you, Marcel. Thank you so much for being here. Moving to Melissa Belen Gonzalez. Melissa.
Hi, good morning, everyone. I'm a Marine Education Fellow, just like Isaac, at the UGA Marine Education Center and Aquarium. We're very much looking forward to summer camps. We've done a lot of teaching virtually, a lot of outdoor things, but now we're getting back into in-person teaching. We've turned some of our little projects into lesson plans that we'll also be testing out for summer camps. But yeah, it's nice to see everyone across all the different departments. I know it's been really hard during COVID, so.
Thanks, Melissa. And thanks to all our Marine Education Fellows for doing all that they do. Thanks for being our ambassadors. Moving to Natalia Jousting.
Hi, everybody. I'm Natalia. Just like Melissa, Isaac, and Emily, I'm also a Marine Education Fellow. And my research interests include the fisheries, and I am really interested in specific movement patterns of certain fish.
Fabulous. I think I've got through everybody on my list. I would like to now invite anyone who hasn't had a chance to introduce themselves. If you are one of the lightning round speakers, I skipped you because you'll get a chance to do so. If you're one of our panelists, I skipped you because you will get a chance to do so. But if you're not a lightning round speaker or one of our panelists today, please go ahead and introduce yourself.
I spoke yesterday, but I didn't introduce myself.
Please
I'm
go
Mark
ahead.
First year at the Skidway Institute of Oceanography. I'm a biological oceanography and microbial ecologist. Really fascinated. It sort of motivates me is the interactions between microbes and other microbes, their environment, and especially bigger stuff, which I talked about shrimp blackgill yesterday, which is a microbe-macrobe interaction, a parasite in a larger organism. And I've been involved in Sea Grant for a while, and it's always just so inspiring to hear the breadth and depth of everything that's going on. Gotten a few ideas, and hey, I'll be contacting some people. So thank you, everybody, and that's me.
Great. Thank you, Mark, for all that you do and your contributions to our program. Anyone else?
I can jump in real quick.
Alexandra, please go ahead.
Yeah, hi, I'm Alex Miscalis. I'm a PhD student at Georgia Tech. I work with Kevin Haas, and I'm in the Ocean Science and Engineering program. My interests mostly lie in coastal engineering, and within that, I'm especially looking at shipwake and nature-based resiliency these days. Thank you.
Lovely to see you, Alexandra. Thank you so much. I heard somebody else too.
It was me, Nina.
Go ahead, Nina.
I gave a lightning round talk yesterday too, but wanted to introduce myself to everybody. I'm Nina Sassano. I coordinate the intern program here at the UGA Marine Extension, Georgia Sea Grant. I oversee a lot of our student opportunities. So if you are a student or have any interest in working with students, contact me and we can make some magic happen. Nice to meet everybody.
Thank you, Nina, for all that you do for our program. Anyone else? All right, I think we're ready to dive back into the presentations, Kimberly. I don't think Kun is here as yet, so we'll wait for her until the end, but we'll move on to the next speaker. So I'll start projecting again and mute myself.
Okay, to kick us off for our final round of our lightning talks, we're going to have Shannon Matsky.
Thank you. Yes, I am Shannon Matsky. I am a master's student at Georgia Southern and a current Georgia Sea Grant research trainee. And the research I'm talking about is also part of a larger SIG funded project. So, coastal living. It's a dream of people around the world. And in fact, 40% of the world's population lives within 60 miles of a coastline. And here in Georgia, we're lucky to have a beautiful coast that includes the beaches of Tybee Island. Tybee is Georgia's most developed beach, and it is also a hub of tourism each summer. Historically, a sheltered coast, Tybee has been hit with multiple extreme storms in the last decade. Next slide. And these storms are only going to become stronger and more frequent as a result of human-induced climate change. And last year, Tybee began construction on a series of coastal sand dunes to provide protection from future storms. Next slide. Coastal sand dunes have been shown to weaken the force of ocean waves during storms, and in particular, vegetated sand dunes provide 40% more of a reduction of erosion than dunes with no vegetation. Below ground networks of roots and rhizomes bind sand to stabilize dunes, and above ground structures trap sand to continue dune building. So imagine a windy day on a beach. You might notice grains of sand blowing in the wind inland and away from the ocean. These grains will ride on the breeze until they hit a structure like the blade of a dune grass. The grain of sand will then drop onto the dune. So imagine that this happens to thousands of grains of sand every second. You can see that vegetation plays a vital role in sand accumulation on sand dunes. But are there certain species of plants that can trap sand or stabilize dunes better than others? And is there an optimum number of plants on a dune that balances financial cost with ecological function? That is what I am researching. Next slide, please. My study aims to both monitor the success of the dunes that Tybee created and to better understand the most effective planting strategies for dune growth and its stability. I'm doing this by comparing the growth and survival of planted vegetation, as well as sand accumulation, in sites of high and low density plantings versus that of a control area of bare dune where no vegetation was planted. And you can see that here in this drone image, the areas of low, high density, and then a bare site with no vegetation. So the overall goal of Tybee's dune construction is to protect coastal development and residents. And my experiment can add extra layers to this project because I'm working to determine what the best practices are in regards to planting this dune vegetation. So I'll be able to provide data to help managers make informed decisions as to which plants and in which densities best suit their management needs of their specific coastal area. And additionally, after every restoration project should come a period of monitoring the success of the overall project. So I'm also looking for correlations between plant growth measurements and sand accumulation so that we can determine which of these plant growth measurements tell us the most about the success of the restoration and the growth and stability of these sand dunes so that these measurements can be taken in the future on Tybee. So my study will provide valuable information on how to best manage Tybee sand dunes for the protection of the island, but the study is also applicable to other coastal areas in need of restoration because we can find out the best practices for both the planting and the monitoring of these constructed sand dunes. Thank you.
Thank you so much, Shannon. Next up, we have Sergio Sabat.
Hi, everyone. I'm losing a little bit of my voice, so I apologize for that. I want to say a special good morning to John Carroll. I'll be sending you my thesis soon. I'm Sergio. I'm a master's student at Georgia Southern University. I'm studying the effects of hydrology on aquatic macroinvertebrates, but also I'm the Georgia Sea Grant State Fellow, in which I'm working with Jekyll Island Authority as well as Georgia Audubon on cultivating strategies and delivering opportunities where birds and people can thrive along the Georgia coastline. So a little bit outside of my area of understanding as I started. For the next slide, please. Yeah, so in terms of birds, especially shorebirds, my focus is since it's a deeply challenging work, I aspire to contribute and voice the need to reserve spaces for these migratory shorebirds, as they not only bring in beauty and ecosystem value to, especially at least in my area of interest, are along the Jekyll Island coast. These shorebirds find refuge as migratory routes, especially the piping plover, which is the small bird you see right in the top left corner. That's one of my central focuses right now, is helping Jekyll Island develop and set up monitoring for these specific piping plovers, but also conduct and improve their current Wilson's plover monitoring, especially their nesting sites, as well as conduct their international shorebird surveys throughout. In regards to Georgia Audubon, with their message of trying to explore and expand areas where people and birds can unite. I'm helping them expand their current programs from Atlanta all the way into the coastline to better, again, support that message. For the next slide. Well, again, I started studying only macroinvertebrates. It's been quite the journey and very interesting. I've been able to meet some cool friends on the route. I am deeply astonished by the work that's currently being done in the coast, and I'm very, very grateful that Mona and everyone else was allowed me the opportunity to be a Georgia Sea Grant Fellow. And I think with that, I end.
Thank you so much, Sergio. Next up, Doug Sampson.
Good morning, everyone. I want to use my time here to highlight some of the major programs, projects, and activities that the Reserve is engaged in right now or will be engaged in the near future. Next slide, please. Our education program, Adam McKinnon is deep into the process of hiring a new assistant education coordinator. This person will be involved in our student educational programming, our teacher workshops, and our group tours. We hope to have them on board this summer in time for the fall semester. Over at Stewardship, Suzanne Van Pererin is leading a team that's doing a major renovation of the boardwalks along our 25-year-old nature trail out by Nantigoat Beach. Rachel Guy is very busy, research coordinator. She's been doing a great job mentoring Sydney Williams, our first Davidson Fellow, who's not quite to the end of her first year with us. The National NEARS Network will be announcing the second round of Davidson Graduate Fellowships next month. Stay tuned for that announcement. Nate Nevelink mentioned the Estuarine Fish Monitoring Cooperative yesterday that Rachel developed with partners at UGA, the Marine Institute, Sea Grant, and College of Coastal Georgia. That's a big, exciting project. Rachel is also involved in 2 NEARS Science Collaborative projects with other reserves along the southeast coast. Next slide, please. Facilities-wise, the island manager, Fred Hay, has overseen a major renovation and enhancement of the Sapelo Island Lighthouse. Last year was the 200th anniversary of the lighthouse, and we had hoped to do a big celebration that COVID precluded. Back on the mainland, we hope to find money next year for a major expansion and renovation of the Sapelo Island Visitors Center, which is also now 25 years old and needs a makeover. Administratively, I'm really pleased that we finally submitted our management plan to NOAA. We hope to hear back from them in the next month or so. And then we're also working with Simona Perry on a new communications plan for the reserve, which will be our first ever plan. Next slide. Thank you.
Thank you, Doug. Next up, we have Marshall Telefert.
Oh, the color is red. I was surprised. Okay, so I'm here to talk about our project. This is a collaboration mainly with my students, Emma Livernois, Evan Maguette, and then also Nicole Lopanic at Georgia Tech and Katie Smith at the Water Quality Program in Brunswick. And I also want to acknowledge these people at the bottom there that basically always help us out when we come down to Skidaway or work around the coast. Next slide, please. So I don't want to spend too much time on this. Most people know that atmospheric CO2 has been contributing to acidification of the oceans, obviously with some important applications on calcifying organisms. The 2 reactions are shown on the bottom left here, and also phytoplankton growth. Which has, of course, important repercussions on fisheries and, of course, economic impact. Now, those processes are relatively well known in the open oceans, although some quantitative aspects of it remain still questions. But in coastal environments, the effect of rivers and then sediments is not well characterized. And so we know that the rivers provide significant amount of minerals, carbonate minerals and clays that can contribute to mitigating ocean acidification by generating alkalinity. But the sediments generate both acid and alkaline reserve. And so it's pretty difficult to quantify, to figure out if that contributes to ocean acidification or mitigate or help mitigate ocean acidification. So this is what this project is about, determining the role of sediments in coastal areas on the flux of alkalinity and DIC, or dissolving organic carbon, in the sediments. Next slide. And so to realize, the next slide, please. Thank you. So to realize this objective, we use actually benthic landers that's shown here on the picture on the left. We deploy these chambers on the seafloor to measure the flux as a function of time in the overlying water above the sediment. And as you can see on these graphs here, these two graphs stacked on the top of each other as a function of time shows dissolving organic carbon and alkalinity as a function of time. And we for this project, we identified two areas where we want to investigate these processes, basically in Savannah, because it's an urban environment that's exposed to nutrient inputs from anthropogenic activities, and then near Sapelo, where the environment is much more pristine, to try to see what the differences are in terms of these benthic flux of alkalinity and CO2. And as you can see from looking at these graphs, in Savannah, the fluxes are much more important, although overall both fluxes seems to be relatively low from these sediments. And so we try to figure out now, using these benthic landers, we also have a micromanipulator and sensors that inserted in the sediments over depth can help us figure out what are the processes responsible for the generation of acidity and alkalinity. And so we try to figure out the reasons behind these differences. What is interesting, and I'll finish with that, is that the Savannah sites, if you look in the right column of that table, the ratio of alkalinity to DIC is pretty close to 1, which suggests that in fact a significant input of alkalinity may actually not be negative for the overlying water. And so we think that denitrification by the supply of nutrients in this particular area is probably generating this alkalinity, which is kind of interesting because nutrients are typically considered to be a pollutant. In this case, these nutrients have a positive effect on ocean or positive effect on ocean acidification, meaning that it's a good effect. I'll stop here. Thank you.
Thank you very much, Marshall. And to bring us in, we're going to end with Dr. Jacques Kelly.
Hi, everybody. I'm Jackie Kelly. I'm going to give a presentation for Jessica Ware, who's an undergraduate student who's working on me. She's at field camp right now, which is the capstone experience for our degree. And so she was a Georgia Sea Grant undergraduate trainee fellowship for this past year. And she was looking at, sorry about the background noise again. She was looking at groundwater ages underneath of the salt marsh near St. Simons Island, Georgia. And this marsh was experiencing dieback. It's a marsh site that I've been monitoring with Dr. Christine Laddick since 2014. So the bottom picture shows the dieback in the marsh site here. And so next slide, please. So the primary purpose of this project is to really understand if groundwater residence time, that's how long the groundwater stays underneath of the marsh, is impacting the dieback that's happening at this marsh site. That picture from 2014 or 2016 or whatever that was, the marsh looks much better than that now, but there is still some areas of recovery on that field site. So she was using short-lived radium isotopes and radon isotopes, as well as basic water quality parameters, to try to gauge how long the water was staying underneath of the marsh. The next slide, please. And so she concluded that in the 1-year study that she was doing on the field site, that there was not really any relationship that she could tease out between the health of the Spartina alternative flora on that field site and the connection with how long the groundwater is staying underneath the field site. But it was just limited in scope, so we're expanding that to go back to some archival data that I've collected. Now that we've got the methodology worked out for figuring out how old the groundwater is underneath of this marsh site. And so, I'm sorry. And so I'll just, I'll end with that because I better go. But thank you for the presentation, and take care, okay?
Thank you, Jackie, for your research and your wonderful motherhood.
That was fabulous. We finally have time for a little bio break, everyone. We will resume the rest of the program at 10 a.m. So Bonnie, may I request you to drop a link for the webinar, please, so that we can get started promptly at 10 a.m. Thank you so much for facilitating this session, Kimberly. Really appreciate it. So Bonnie, if you can drop the link, I think that will be it. And while Bonnie is doing that, let's wish Mark a very happy birthday. Mark, wish you happy birthday. Also to Anne today.
Thank you, Mona.
Happy birthday. Thank you all for coming. See you on the next Zoom.